There's a question buried inside the human genome that scientists spent decades figuring out how to ask properly. Not just how old is our species, and not just where did we come from, but which living populations carry the oldest DNA, the genetic lineages that stretch back furthest in time without interruption, the branches of the human family tree that have been growing the longest without being pruned or grafted or replaced. It sounds like a simple question. It turns out to be one of the most complicated and one of the most revealing that genomics has ever attempted to answer. The answer, when it finally came into focus through years of increasingly sophisticated ancient DNA analysis and population genetics modeling, was not what most people expected. The populations that carry the oldest, most genetically diverse, most deep-rooted DNA on Earth are not the ones that built the pyramids, not the ones who launched the age of exploration, not the ones whose civilizations fill the history books that most of the world grew up reading. The oldest human DNA on Earth belongs to people living in southern Africa, specifically to groups like the San people of the Kalahari and the Kuko of the Cape, populations that most of the world has barely heard of, living in places that most history curricula treat as footnotes. And the implications of that finding, once you sit with them long enough, quietly reorganize almost everything you thought you understood about the hierarchy of human antiquity. But to understand why that answer is what it is, you have to first understand what scientists actually mean when they talk about the age of DNA. Because DNA age, as a scientific concept, is not what popular language tends to imply. It does not mean that one group of people is more evolved than another, or more primitive, or further along some imaginary ladder of biological progress. It does not mean that their DNA is older in the sense of being degraded or archaic. What it means is something more precise and more interesting. It refers to genetic diversity, the number of distinct variants present in a population's genome, and to the depth of lineage, meaning how far back the genealogical branches of a population's genetic tree extend before they converge on common ancestors. Populations with the highest genetic diversity and the deepest, most branching lineage trees are, in this technical sense, the oldest, because they have been accumulating genetic variation for the longest uninterrupted period of time without experiencing the dramatic population bottlenecks that winnow diversity away. And that distinction between populations that have been accumulating diversity continuously and populations that went through severe bottlenecks is the central key to understanding why the ranking looks the way it does. To appreciate what genetic diversity really means in practice, it helps to think about what happens in its absence. When populations go through bottlenecks, when their numbers crash to very small counts, whether due to migration, famine, disease, or geographic isolation, large portions of the genetic variation that existed before the bottleneck simply disappear. Random chance determines which variants survive and which are lost forever. This process, known as genetic drift, is ruthless and indiscriminate. It does not select for useful traits or eliminate harmful ones. It simply reduces the palette of variation available to a population and to all of that population's descendants. Every successive bottleneck compounds the effect, narrowing the palette further. Populations that have experienced many bottlenecks over their history carry genomes that are, in a measurable sense, compressed, less diverse, more uniform, less reflective of the full range of variants that our species began with. Populations that avoided those bottlenecks carry genomes that are, in comparison, expansive, varied, and deep. The story begins in Africa, where it always begins when you trace human origins far enough back. Modern Homo sapiens evolved in Africa somewhere between 200,000 and 300,000 years ago, with some contested evidence pushing that estimate even further. For most of that time, for the vast majority of our species' existence, all humans lived in Africa. The populations that remained in Africa after the Great Migration Out, which began roughly 60,000 to 70,000 years ago, are the direct descendants of those original populations, separated by thousands of generations of continuous residence in the continent where our species first emerged. They have had more time to accumulate genetic variants, more time for mutations to arise and spread, and crucially, they did not pass through the severe genetic bottleneck that the out-of-Africa migration imposed on the ancestors of every non-African population on Earth. That bottleneck is the critical concept. When a small group of people, and genetic modeling suggests the founding population that left Africa may have been only a few hundred to a few thousand individuals, breaks away from a larger population and founds a new lineage, they carry with them only a sample of the original group's genetic diversity. Like taking a handful of different colored marbles out of a jar containing thousands, the handful cannot represent the full range of the jar's contents. Every non-African population on Earth is descended from that handful. Every non-African genome carries the imprint of that sampling event, that reduction, that loss. The populations who stayed in Africa, who never went through that bottleneck, who kept drawing from the full jar, retained a depth and breadth of genetic diversity that no non-African population can match. That is what the data says, across every measurement in every data set, every time the question has been asked. The San people of southern Africa represent perhaps the clearest demonstration of this principle. Genetic studies have consistently placed sand individuals and sand populations at the deepest roots of the human genealogical tree, meaning their lineages diverge from those of all other living humans at the earliest points in time. Research published in the journal Science, drawing on complete genome sequences from sand individuals, found that the sand carry mitochondrial DNA lineages, passed directly from mother to child without recombination, that diverge from those of other human populations at dates estimated to be 100,000 years ago or more. Some studies have pushed estimates of the deepest sand lineage divergences to 200,000 years or beyond, figures that approach the very origin point of anatomically modern humans. To put that in perspective, when the ancestors of Europeans and East Asians and Native Americans were all still part of the same ancestral African population, sand lineages had already been diverging for tens of thousands of years. They are not just old, they are foundational. The genetic diversity within sand populations is staggering by any comparative standard. Studies measuring single nucleotide polymorphisms, individual positions in the genome where people differ by a single DNA letter, have found that two unrelated sand individuals from neighboring communities can differ at more genomic positions than a European and an East Asian selected at random from opposite ends of the Eurasian continent. That is not a small effect. That is a reversal of the intuitive expectation that people who look similar and live near each other must be genetically similar to each other. The sand are a living demonstration that visible similarity is a deeply unreliable guide to genetic relatedness or diversity. What makes the sand case even more remarkable is the context in which this diversity exists. These are not large, urban, cosmopolitan populations drawing from many distant lineages through migration and intermarriage. These are relatively small, geographically bounded communities of hunter-gatherers and pastoralists living in one of the harshest environments on Earth, whose social structures have remained remarkably stable across thousands of years. And yet, the genetic variation present within those communities exceeds what you would find if you sampled randomly from the entire landmass of Europe. The implication is not subtle. Isolation does not automatically mean uniformity, and small population size does not automatically mean low diversity when the lineage is old enough and deep enough. The San are carrying in their genomes today the accumulated genetic history of a lineage that was never subjected to the kind of brutal filtration that all non-African lineages passed through. That history is legible, and it is astonishing. The Hadza of Tanzania, a small population of hunter-gatherers whose language, like that of the San, uses click consonants, represent another extraordinarily deep genetic lineage. Their divergence from other African populations appears to have occurred very early in the history of modern humans on the continent. And like the San, they carry levels of within-group genetic diversity that dwarf those of most non-African populations. The Hadza have been a small population for a very long time, which paradoxically makes their retained diversity even more impressive. Population size alone does not predict diversity when the lineage is old enough and continuous enough. Other populations in Africa, the Mbuti and Aka Pygmies of Central Africa, the Sandawe of Tanzania, various groups in West Africa, also represent lineages of extraordinary depth. Africa as a whole contains more genetic diversity than the rest of the world combined. That is not a rhetorical flourish. It is a measured, replicated, well-established finding of population genetics. When scientists sequence genomes from populations across sub-Saharan Africa, they find levels of variation in every measurable dimension, from single letter changes to large structural variants, that make the variation found across all non-African populations look like a regional dialect within a much larger conversation. Europe and Asia and the Americas and Oceania, taken together, represent a genetic subset of African diversity, not its complement. This is a profound implication that rarely gets stated clearly in popular accounts of human genetics. When we talk about racial or ethnic differences in DNA, when we ask whether Chinese DNA differs from European DNA, or whether African DNA is different from either, we are, in every case, asking about differences that are smaller than the differences contained within Africa itself. The continent that is most often treated as peripheral in popular histories of human civilization is, genetically, the center of everything. Every genome on Earth is an African genome, modified to varying degrees by the experiences of populations that left the continent between 40,000 and 70,000 years ago. The further a population's ancestors traveled from Africa, and the more dramatic the bottlenecks they passed through, the less of that original African diversity they carry. The populations that left Africa and traveled furthest, both in terms of geographic distance and in terms of the number of successive founding events and bottlenecks along the way, are, in this genomic sense, the youngest. Indigenous peoples of the Americas offer the most striking example. The founding of the human populations of North and South America appears to have occurred through a bottleneck that was itself downstream of the original out-of-Africa bottleneck. The ancestors of Native American populations are believed to have migrated from a Siberian or northeastern Asian source population, themselves already descended from the out-of-Africa founders, and then crossed into the Americas through Beringia, a land bridge that was exposed during the last glacial maximum and dispersed rapidly through 2 empty continents. Each of these successive steps, out of Africa, across Asia, through Beringia, down through the Americas, reduced genetic diversity further. The result is that many indigenous American populations show among the lowest levels of genetic diversity of any population on Earth, not because there is anything inferior about their genomes, but because their ancestors passed through more successive population bottlenecks than almost any other group of humans alive today. They are, in the precise technical sense, the youngest DNA, not old in the chronological sense of being archaic, but young in the evolutionary sense of having the least accumulated variation, the shallowest lineage trees. It is important to pause here and appreciate what this means for how we think about indigenous American populations more broadly. These are peoples with extraordinarily rich and complex cultures, languages, spiritual traditions, agricultural innovations, and civilizational achievements. The Aztec, the Maya, the Inca, the Haudenosaunee, the Lakota, all built worlds of extraordinary sophistication. Their relative genetic youth, in the population genomic sense, says absolutely nothing about the depth, complexity, or value of their cultural heritage. Genetics and culture operate on entirely different timescales and through entirely different mechanisms. A population can have the most recently founded genome on Earth and the oldest living oral traditions, the youngest genetic lineage, and the most sophisticated astronomical knowledge. These dimensions do not speak to each other. What the genomic finding tells us is only about the history of migration and population founding, not about the people themselves. The populations of Oceania, Aboriginal Australians and Papuans from New Guinea, occupy a complicated and fascinating position in this ranking. Their ancestors left Africa early and traveled an almost incomprehensibly long distance through southern Asia, before crossing into Australia and New Guinea at a time when sea levels were lower and the distances involved, while still formidable, were shorter than they would become. Aboriginal Australians have been living on their continent in remarkable genetic isolation for somewhere between 50,000 and 65,000 years, longer than any other population outside Africa. In terms of their separation from other living human populations, they are extraordinarily ancient. In terms of their within-group diversity, they fall below African populations, but retain diversity that reflects both their early departure from Africa and their long period of isolated accumulation. Aboriginal Australian mitochondrial lineages represent some of the earliest branching non-African lineages in the human genealogical tree. They also carry, as noted in other comparative genomic contexts, the highest levels of Denisovan ancestry of any living population, a consequence of their ancestors' early passage through Southeast Asia, where Denisovan populations apparently existed in greater numbers than anywhere else. The Denisovan component of Aboriginal Australian and Papuan ancestry is itself a reminder of how complex the human story actually is. Denisovans were a separate archaic human lineage, distinct from both Neanderthals and modern Homo sapiens, who apparently occupied parts of Asia for hundreds of thousands of years before the arrival of modern humans. The ancestors of today's Aboriginal Australians interbred with these people. That genetic legacy, still detectable today in the genomes of people living in Australia and Papua New Guinea, represents another layer of depth, another connection to an even older chapter of the human story that no other living population carries in the same concentration. The question of where specific Asian and European populations fall in this ranking is more nuanced and more politically charged than the African story, and the genomic findings are correspondingly more complex. East Asian populations, including Han Chinese, show intermediate levels of genetic diversity, well below African populations, somewhat above European populations in certain measures, and reflecting the complex mix of an early out-of-Africa founding, subsequent adaptation, and the absorption of multiple waves of both local and more distant ancestry. Japanese populations, particularly the indigenous Ainu of Hokkaido, carry genetic signatures of ancient lineages distinct from those of the mainland Han populations, adding further depth to East Asian genomic complexity. European populations show the effects of the particularly dramatic demographic history described in other genomic comparisons. The near-total replacement of the original European hunter-gatherer populations, first by Anatolian farmers, and then by steppe pastoralists, meaning that the genetic lineages currently dominant in Europe are substantially younger, in the sense of having been present in Europe for a shorter time than the lineages currently dominant in Africa or even in much of East Asia. This does not mean Europeans are less human or less interesting or less ancient as a species. It means that the specific genetic ancestry now characteristic of European populations arrived in Europe relatively recently, carried by migrants who brought it from other places. The deep hunter-gatherer ancestry that predates those migrations, the remnants of the people who painted caves in southern France and northern Spain, is still present in European genomes, but it is a minority component. The European genome is not a particularly ancient one in the population genetic sense, even though the territory of Europe has been inhabited by modern humans for more than 40,000 years. The story of those cave painters deserves a moment of consideration. The people who created the extraordinary art at Lascaux and Altamira and Chauvet, the horses, the aurochs, the handprints pressed against stone walls and the flickering light of animal fat lamps, were genetically quite distinct from the populations that now inhabit those same landscapes. They were European hunter-gatherers of the Upper Paleolithic, carrying their own distinctive genetic profile, their own mitochondrial and Y chromosome lineages. When the Anatolian farmers arrived around 9,000 years ago, and when the steppe pastoralists followed around 5,000 years ago, the genetic legacy of those cave artists was diluted, pushed to the margins, replaced in the majority by the newcomers' lineages. Europeans today carry a fraction of that ancient ancestry, perhaps 10 to 15% in Western European populations, somewhat more in isolated pockets like Scandinavia or the Basque Country. The rest arrived later. The genetic European, in other words, is in many ways a relatively recent composite, assembled from sources that were themselves already products of the out-of-Africa bottleneck and its subsequent regional history. South Asian populations, the people of the Indian subcontinent, represent one of the most genetically diverse non-African regions on Earth. India's population is the product of at least two major admixture events, the mixing of ancient South Asian hunter-gatherers with a wave of West Eurasian farmers who arrived from the northwest, followed by a second wave of steppe ancestry that brought the ancestors of the Indo-Aryan languages into the subcontinent around 4,000 years ago. The result is a genetic patchwork of extraordinary richness, with populations in southern India carrying significantly higher proportions of the ancient local ancestry, and populations in the north carrying more of the steppe and West Eurasian components. Some of the ancient South Asian lineages, the ancestors of today's indigenous tribal populations and southern Dravidian groups, are among the earliest branching non-African lineages outside Australia. The populations of the Middle East and North Africa occupy yet another complicated position in this landscape. The Fertile Crescent, the region that gave the world its first cities, its first writing systems, its first large-scale agricultural societies, is, from a genomic perspective, a region of ancient crossroads rather than deep indigenous isolation. Its populations reflect millennia of movement, trade, conquest, and intermarriage between African, Eurasian, and local lineages. The result is a mosaic of intermediate diversity, deeper than the populations of far northern Europe or the Americas, shallower than the populations of sub-Saharan Africa, and rich with the complex signatures of repeated admixture events. The people who first wrote things down, who first built walls and granaries and temples, were not genomically ancient in the way that the San or the Hadza are ancient. They were, in the genetic sense, already composites, products of thousands of years of mixing between the descendants of multiple out-of-Africa founding events. What the ranking ultimately reveals, when you look at it honestly and in full, is that the hierarchy human societies have historically imposed on populations, with literate, technologically complex civilizations at the top and hunter-gatherer societies at the bottom, is precisely inverted by the genomic evidence. The populations that built no pyramids, launched no navies, and left no written records are, by the measure of DNA age and depth, the most ancient and most genetically complex humans alive. The San, the Hadza, the Mbuti, the Sandawe, people who have been described in colonial-era literature as primitive, as stuck in the past, as representatives of humanity's childhood, are, in the genomic record, humanity's elders. Their DNA contains variation that was already ancient when the first city was built in Mesopotamia. Their lineages were already diverging from those of other humans, while the ancestors of every other population on Earth were still part of the same ancestral African community. That is not a minor adjustment to the popular understanding of human history. It is a structural inversion of it. The populations that history has treated as marginal are, in the most rigorous scientific sense, foundational. The populations that history has placed at the center, the great civilizational powers of Europe and Asia, are, genetically speaking, derivative. They are branches from a tree whose deepest roots run through the Kalahari and the Congo Basin, not through the Nile Valley or the Yellow River or the Rhine. None of this should be interpreted as a ranking of human worth, intelligence, cultural sophistication, or civilizational achievement. Genetic age is not a measure of human value. A younger genome is not a lesser genome. The bottlenecks that reduced genetic diversity in non-African populations were accidents of geography and migration, not judgments of merit. The San people are not genetically superior to Europeans or East Asians because their lineages are older. They are not more human, not more biologically interesting, not more deserving of anything. They are simply the living population whose ancestors stayed closest to the origin point the longest. who did not go through the filter that reduced everyone else. That is a fact about history and geography, not a fact about human hierarchy. There's one more dimension to this story that deserves to be named directly, because it has practical consequences in the world today. The same populations that the genomic record identifies as humanity's eldest, the San, the Hadza, the Mbuti, the Aboriginal Australians, the indigenous peoples of the Amazon, are also, in the political and economic realities of the present moment, among the most dispossessed and endangered. Their lands are under pressure from extractive industries and agricultural expansion. Their languages are dying at rates that alarm linguists. Their children are being pulled into urban economies that do not recognize or value the knowledge systems their communities have accumulated across thousands of generations. The irony is almost too large to hold. The people whose DNA contains the most ancient and irreplaceable record of human genetic history are the same people whose living cultures are most at risk of being erased by the economic and political forces of the modern world. Preserving those cultures is not just a matter of justice, though it is certainly that. It is a matter of preserving something that, once lost, cannot be recovered. A depth of genetic and cultural continuity that took 200,000 years to build and that the current century is actively threatening to destroy in a matter of decades. What the science is patient about, what it keeps demonstrating in study after study with increasingly refined tools and increasingly large data sets, is that the story of human genetic age runs exactly counter to the story of human cultural dominance that the last few centuries have told. The genomes do not care about empires. They do not care about literacy rates or GDP or military power. They keep their own record, written in mutations that accumulate at a steady, predictable rate, and that record says clearly and repeatedly, the deepest human past lives in Africa. The oldest human DNA belongs to people who have been in place the longest, who went through the fewest bottlenecks, who retained the most of what our species began with. Everything else, every civilization, every religion, every language, every empire that has risen and fallen since, is built on a genetic foundation that was already ancient when recorded history began. The oldest DNA on Earth does not belong to the people who wrote the history books. It belongs to the people who were never in them. And that, more than any other finding in modern genomics, should give us pause about what we think we know, and what we still have left to understand. The genome is the longest book ever written. It has no author, no publisher, and no agenda. It simply records, in the language of mutation and inheritance, where we came from, what we went through, and which among us has been here the longest. When you learn to read that language, when you understand what the data is actually saying, the world looks different than it did before. Not because the people in it have changed, but because you have finally begun to see them clearly.